This is the first introductory video on the topic of cancer, a complex group of diseases which afflicts people of all ages but becomes more prevalent in older age. Furthermore, it is not restricted to humans, but occurs in all animals, although it is rare in some animals such as elephants, porpoises, and whales, a fact that will be reviewed in a later video. As mentioned, cancer is not a unitary disease, but is the common name for a multiplicity of diseases, all of whom are characterized by uncontrolled cell growth and division. This abnormal growth is caused by changes in the cell's DNA, called mutations. These have many causes or etiologies. They may be genetical, inherited from our parents, or acquired throughout our lives due to exposure to carcinogens, cancer-causing agents like tobacco smoke and radiation. Cancer can develop in almost any part of the body. It disrupts the normal functioning of organs and tissues by releasing chemicals which may disrupt homeostasis, preventing nutrients reaching healthy cells by directing more supplies to themselves. Understanding the causes and mechanisms of cancer development is crucial for effective prevention, diagnosis, and treatment. The human body is made up of trillions of cells, each programmed to grow, divide, and die in an orderly manner. Cancer arises when a cell undergoes a mutation, changing it to become the cancer-initiating cell. This cell loses its programmed control, multiplying rapidly and invading surrounding tissues. There are two types of mutations associated with cancerous cells. Specific cancer mutations are what change a cell into a cancerous cell, that is, a cell that does not stop dividing and replicating. This causes genetic instability in that cell, resulting in further tumor progression mutations, which in turn accelerate the rate of growth and suppression of regulatory pathways. This uncontrolled growth can lead to the formation of a mass of abnormal cells called a tumor, which will contain cancer stem cells. These cells have reverted in their biochemistry to represent certain characteristics of normal stem cells, such as the ability to differentiate into the cell types within the developing tumor. They are the main agents for the progression of cancer. Oncogenes are a group of genes that play a critical role in the development of cancer. These genes are derived from proto-oncogenes such as RAS, WNT, MIC, TRK, ARC, which code for proteins involved in cell growth and division. Once mutated to oncogenes, they will overexpress and overproduce these proteins, thus disrupting the Hayflick limit. Named after the American anatomist Leonard Hayflick, who discovered that there is a natural barrier that limits the number of times a normal cell can divide. This limit is regulated by telomeres, protective caps at the ends of chromosomes that shorten with each cell division. Once telomeres become too short, the cell enters a state of senescence and stops dividing. Cancer cells, however, have developed mechanisms to bypass the Hayflick limit. Mutations in oncogenes, namely the TERT promoter mutation, which produces a binding motif responsible for nullifying the repression of TERT gene expression, a gene that can activate telomerase production. Telomerase is an enzyme that can lengthen telomeres, allowing cancer cells to divide indefinitely. This immortalization is a hallmark of cancer and contributes to its uncontrolled growth. Cancer is broadly classified into different types based on the type of cell from which it originates. The most common types of cancer include carcinomas, sarcomas, lymphomas, leukemias, and myelomas. Carcinomas are cancers that arise from epithelial cells, which are the cells that line the surfaces of the body and internal organs. Examples of carcinomas include lung cancer, breast cancer, and colorectal cancer. Sarcomas originate from connective tissues such as bones, muscles, and cartilage. Examples include osteosarcoma, the most common cancer of the bones. Lymphomas and leukemias are cancers of the blood and bone marrow, while myelomas affect plasma cells, a type of white blood cell. Each type of cancer has its own unique characteristics, growth patterns, and treatment approaches, which will be discussed in future videos of this series. Understanding these differences is crucial for developing effective treatment strategies. Not all tumors are cancerous. Tumors can be classified as either benign or malignant. The American National Institute of Health surmised that benign tumor cells are due to 2 or 3 specific cancer mutations, whereas malignant tumor cells contain 4 specific cancer mutations and 1 to 3 tumor progression mutations. Benign tumors are non-cancerous growths that are typically slow-growing and do not spread to other parts of the body. They are often encapsulated, meaning they are surrounded by a layer of tissue that separates them from surrounding structures. Malignant tumors, on the other hand, are cancerous. They have the ability to invade nearby tissues and spread to distant sites through a process called metastasis. Malignant tumors can grow rapidly and are more likely to be life-threatening. The distinction between benign and malignant tumors is crucial for determining the appropriate course of treatment. While benign tumors may only require monitoring or surgical removal, malignant tumors often necessitate more aggressive treatments such as chemotherapy, radiation therapy, and immunotherapy. We hope that this introductory video in our series on cancer was informative. If so, please hit the like button, subscribe to this channel, hit the notification bell to be alerted to future videos uploaded. Most importantly, stay healthy and curious.